Hi, my name is Pourya Naderi. I'm a postdoctoral fellow at the Hyde Lab at Beth Israel Deaconess Medical Center and Harvard Medical School. Today, I will be presenting our work on capturing microRNA-controlled coordinated pathway activity, or CARAWAY. MicroRNAs are potent post-transcriptional regulators of gene expression, and each microRNA may target up to hundreds of genes that span across multiple pathways at the same time. However, detecting key microRNA regulators that can coordinate transcriptional programs across multiple pathways is still an open problem. CARAWAY is a four-stage pipeline that identifies groups of coordinated pathways and returns microRNAs that concurrently regulate them. In particular, CARAWAY takes a gene expression dataset as input and initially maps the gene expression to pathway summary statistics. These pathway summary statistics indicate the activity of known pathways across different samples. We then take pathway summary statistics to identify differential pathway summaries, or DE pathways, between case and control. To identify clusters of co-regulated DE pathways, we use PCXN tools, which is based on canonical correlation between known pathways. In the last step, CARAWAY returns a list of microRNA and p-values. In particular, given a group of pathways P1 to Pm, we take the p-value of enrichment between the microRNA and each one of the pathways within the group. We transform them via inverse normal transformation, and we summarize these transform scores across the group of pathways. We then generate the p-value for CARAWAY using empirical distribution for random M pathways across the universe of pathways. We applied CARAWAY to discover microRNAs coordinating resilience against Alzheimer's disease in individuals who have pathological features of AD but no dementia. In particular, we used 255 samples from the RoseMap dataset, and to generate pathway summaries, we use MSeqDB annotated pathways. So here in this figure, you can see the clusters of pathways that were queried from PCXN. In particular, we see two groups. One is upregulated pathways, pathways that are up in AD, and pathways that are down in AD. And we see the correlation between the pathways. Red denotes positive correlation between pathways, and blue denotes negative correlation. So we applied microRNA prioritization part of CARAWAY, and we evaluated the readouts from this section in two methods. We took a data-driven approach to validate the readouts of CARAWAY. In particular, we used RoseMap microRNA expression dataset and queried which microRNA expression correlated with the activity of pathways within each cluster. We hypothesized that the microRNAs that correlate the most with the pathways have significant overlap with the readouts from the targeting approach from CARAWAY. Here in Table 1, we display the top microRNA that have most correlation with the pathways in the upregulated cluster of AD resilience. The ones that are highlighted in yellow denote the microRNAs that were also prioritized by the targeting approach of CARAWAY. We also see the overlap between the microRNA prioritization of CARAWAY and the correlation is statistically significant. On the other hand, when we use standard enrichment analysis and meta-analysis approaches, we don't see any significant overlap. Particularly, this is because of biases in existing standard methods. We show that standard enrichment analysis and meta-analysis methods tend to produce higher ranks and smaller p-values for the microRNAs that have larger number of targets. As you can see in the left panel in the figure 2, in the x-axis, we have the log number of targets for a microRNA. Each point represents a microRNA, and the y-axis represents the negative log p-value that are generated by the meta-analysis. As you can see here, microRNAs with larger number of targets have larger negative log p-values, and tend to be prioritized in the meta-analysis. As shown in the right panel of the figure 2, CARAWAY is able to bypass this bias towards the number of targets and is able to prioritize microRNAs even when they have small number of targets. In summary, we have shown CARAWAY is an unbiased and sensitive multipathway microRNA prioritization method, and we show that by applying that to AD resilience dataset, we are able to identify key microRNA regulators. You can check out the source code for CARAWAY in the link provided below. Thank you very much.